Chapter 9 deals with controlling the microbial growth in the environment. There's a lot of different terminology that we use, and you may initially think, oh, well, these words are interchangeable, but to a microbiologist, they are not. Antisepsis is going to be using something that's going to reduce the number of microorganisms on living tissue. You don't use an antiseptic on a countertop, etc. You only use it on living tissue versus, say, a disinfectant, which is trying to destroy and remove the microorganisms on non-living tissue. Sanitation is removing microorganisms, specifically the pathogens, from objects and getting it to a level so that it meets your public health standards. So it's not necessarily sterilizing it, but it's reducing the number. Anything that ends with "-side" or "-sidal", basically you're killing the organism. If it ends, such as a bactericidal agent, is going to be killing the bacteria. But if it ends in stasis or static, a bacterial static, it's not killing it. It's going to reduce the numbers and inhibit the growth, but not necessarily kill it. So go over this table and learn the differences in it. Like I said, sanitation and sterilization are not the same thing. Pasteurization and sterilization are not the same thing. This is just showing a graph showing the number of the living microorganisms over time and how the idea of what you're doing when you add some type of an agent is you're reducing the number of microorganisms. How? Because you're killing them, hopefully at a constant rate. What is the action of these various agents, antimicrobial agents, meaning how do they work? Well, there's several different ways that they can work. Sometimes they can alter the cell wall or the membrane of the microorganism. Remember that cell wall is helping to hold the cell together. It's helping to maintain the integrity. And it helps to reduce the effects of changes in osmolarity, changes in pressure of water. So if you damage that cell wall, you're going to make the organism much more susceptible to changes in water pressure, things like that. Sometimes the cytoplasmic membrane helps to, as you know, control what can move in and out. Well, if you damage the cell wall or if you've damaged that membrane, then items inside can leak out because it has been damaged. Non-enveloped viruses tend to have a much higher tolerance for harsher conditions. You can damage proteins, you can damage the nucleic acids. Protein, remember, for them to be functional, structure determines function. So if you damage the structure of it, you will damage the function of it. So some chemicals will denature proteins, which simply means that you have altered the protein's three-dimensional structure, so it's no longer functional. There are some things such as chemicals and radiation and heat that can also alter or destroy the nucleic acids, the DNA, the RNA, and this can result in mutants that become fatal. Whenever you're looking for some, magic new, let's say a disinfectant that you want to use, sorry about that delay. I have a cat that has decided to play. What I was saying was that the microbial control agents, when you're trying to find something new, especially from an industrial standpoint, you're trying to develop something new, certainly you want something that's inexpensive. I mean, we're all consumers. You're not going to buy something if it's real expensive. You want it to work fast. You don't want it to kill the microorganisms a month from now. You want to kill them now. So it needs to be fast acting. You want it to be fairly stable. You don't want to have to, every 2 weeks, go out and buy more, disinfectant. You want to be able to use it. And then when you need to use it again, you go pull it out of your cupboard or wherever you store it. You want it still to be working, so you want it to remain stable. And of course, you want it to be able to control the microbial growth, whether it's killing bacteria, fungi, viruses, whatever. But you don't want it to be harmful to you. You don't want it to be harmful to your pets. So you don't want it to be harmful to other animals. And you don't want it to be harmful to objects. In other words, you don't want it to be breaking down or destroying whatever, let's say it's a spray that you spray it on. So you've got to look at all these things. Sometimes in industry, they may find something that's like, oh, okay, look, this is inexpensive, it works, it's stable, it's fast acting, it's not harmful to humans. Oh, but it destroys plastic on contact, or it destroys countertops on contact. We can't use this item then. In terms of looking at how efficient, when you look at the efficacy of a microbial methods, how efficient is it? You have to look at several different things. Number one, what is the site that's going to be treated? If it's a very harsh chemical, you cannot use it on humans. If it's, say, using extreme heat, we use high heat to, when we autoclave, we use to sterilize certain things. If it's a fragile substance, you cannot use high heat on it. It's going basically to melt it, like plastic petri dishes. When I first started in this field, we'll just say X number of years ago, there were no plastic petri dishes. We used glass petri dishes, and we used to have to, when you were done with it, you autoclaved it. You had to wash them out. You talk about doing dishes. Oh my goodness. We had to wash every one of those out and then sterilize them again. You'd sterilize them when you were done. You'd pour all the fluid, the waste product out, wash them, clean them up. Then you had to sterilize them again. And then you made the new media and poured it in there. You're always washing dishes. The plastic petri dishes, you cannot put those in an autoclave and expect to reuse them. They're going to, trust me, I've seen someone try to do it. You end up with a big blob of molten plastic. It is a mess. So you have to look at the site that's to be treated. Can you use high heat? Not if it's fragile. Can you use harsh chemicals? Not if it's your skin surface. So the method of microbial control is going to be based on what is the site that needs to be treated. This just gives you an idea of how different organisms have different levels of susceptibility to microbial agents, antimicrobial agents. And so the most susceptible, as you can see, are the envelope viruses and the most gram-positive bacteria, and then it moves up. Endospores are some of the most resistant. Prions are very resistant. So you may find something that's effective only against the bottom third or the bottom 2/3. You have to keep that in mind as you're trying to develop something new. What are you trying to kill? What can you use? Different factors that can also affect the efficiency. Like I said, what is the relative susceptibility of the microorganism that you're looking at? And we classify the agents, the germicides. Germicide means that it's going to be killing and they use the term germ because you could say microorganism side, but it's kind of a loud word. High level, intermediate level, and low level. So a high level germicide is going to kill all the pathogens. It's going to include killing those endospores. This thing works. Intermediate level germicides, that will kill the fungal spores, your protozoan cysts, viruses, and pathogenic bacteria, but it's not going to kill those endospores. Low level germicides, now you're just killing the vegetative bacteria, fungi, protozoan, and some viruses. You're not killing any of the spores. So it's not going to be as effective. Once again, you have to look at what type of treatment are you trying to do as to which one of these levels agents you're going to use. Some other things that can affect how efficient your antimicrobial agent is. And the environmental conditions. Temperature and pH can have an effect. You change the pH, number one, that's going to affect the microorganisms' growth. Temperature, typically speaking, the higher the temperature, the greater the death rate, the faster the death rate is going to be. Organic materials, that can interfere with how well an antimicrobial agent is going to work. Sometimes if you have high organic matter, it's going to inactivate some of your chemical disinfectants. Organic materials can interfere with how well, say, heat and chemicals can penetrate, how well radiation can penetrate through your substance. One of the standard things that we tend to use in microbiology is an autoclave to sterilize our substances. General rule of thumb is you need to run it for a minimum of 15 minutes at 15 psi, pounds per square inch, at 121 degrees C. However, the more substances that you are filling up that autoclave, so if you have an autoclave and you're filling it really full, you're going to want to increase the time. If you're putting in, let's say you have test tubes, that's one thing. But what happens if you have a flask that's, say, a 4-liter flask that's filled with 3 liters of broth that you want to sterilize? For that heat to penetrate all the way into the center of that broth and make sure that you actually have sterilized it, you're going to want to extend that time period, because that organic material does interfere with the penetration of the heat. Think of a microwave. You have a big dish. You heat it up in the microwave, and on the surface it might feel hot, but you dig down into your casserole or whatever you're eating. In the middle, it's like, oh, this is still cold. It's kind of the same type thing. This is just showing the effect of temperature on the efficacy of an antimicrobial chemical. So as you can see, the higher temperature, the amount of time that you have to expose that chemical to the organism for it to be effective is much shorter. Cooler temperature, you're going to have to expose it longer. For labs, we have different levels of what we call biosafety levels for classifying labs. There's 4 different levels. Biosafety level 1. This is just very basic lab where you're handling organisms. They do not cause disease in healthy humans. Bio-level safety 2, you're handling moderately hazardous agents. Most of your, say, like high school level labs, if they teach microbiology, would probably be a bio-level safety 1. A lot of your colleges do move up to bio-level safety 2, level 2 labs. Biosafety level 3, you now have to handle the microorganisms in special safety hoods or safety cabinets. Some of your hospitals are going to be a BSL 3. Biosafety level 4 labs. There are only a few of these in the United States. The CDC has one. Several of their labs are level 4s. For a level 4 lab, this is where you are handling those microorganisms that are the most severe. You're working with the worst of the worst ones. You're working with things here that you know can kill. Ebola. You are working with unknown things. If there has been a new outbreak and you don't know what the causative agent is, you're going to be working with in a biosafety level 4, the CDC, with that until you can identify what it is. If it's lethal and has a high contagious rate and there's no cure, that's where you're going to be working with it. There's special training that you have to use in order to work in those labs. This picture shows someone working in a level 4 lab. Notice how they are completely suited up, more than just your lab coat now, folks. You are completely suited up. You have a series of steps, pressure rooms you have to go walk through before you can even enter into this lab. The purpose is it keeps airflow in, pushing everything in. Behind the individual in the back, you can see there is a hood, that thing that kind of has a glass cover. Those are often worked with in a level 2, level 3 labs where the airflow contains and keeps everything in that hood, and there's a special filter in the back. If you're wondering what those yellow tubes are, you can see one hanging from the ceiling on the upper left and then right in the forefront of the picture. Those are respirator tubes. They hook up. If you notice on this individual, there's a yellow tube hooked up to them just below the waist. That hooks up to oxygen supply. So you have your own separate respirator, and you can hook up to the depending on where you are in the room, you can plug in. It does get very tiring, very, very hot wearing that suit. So usually you work in there, and you're not going to spend a full 8-hour shift in there. You're going to work some, leave, come back, work some more, leave, etc. Most of the level 4 ones that I'm familiar with, usually, like I say, you are hooked into a separate airflow. If there was to be a break, contaminant somewhere, you basically usually have a limited amount of time to get out should there be an accident in there. But you notice the boots, the gloves, everything, you're completely sealed in, and then you go through levels of what we call decontamination when you leave the lab. The idea is everything's left in that lab. What are some physical methods for controlling microbial growth? Heat. If you use high temperatures, that's going to denature proteins. If you denature proteins, they're no longer functional. If they're no longer functional, that means all of your various chemical reactions that are dependent on them cannot occur, and that's going to end up killing the cell. High heat can also interfere with your cytoplasmic or cell membrane. It can interfere with the cell wall. It can also, in addition to proteins, it can also disrupt the structure of your nucleic acids. Thermal death point, that is the lowest temperature that's going to kill all the cells in broth in 10 minutes. Thermal death time is the amount of time that's needed to sterilize a set volume of liquid at a set temperature. And so, once again, this is showing that you can measure the microbial death rate and the log amount of time difference it will make. So what do we use in the lab? Moist heat. This is probably one of the most common ones. We use it to disinfect, to sanitize, sterilize, and pasteurize. And remember, from the very beginning table, those are not the same thing. Once again, moist heat is going to help denature proteins. It's going to destroy that cell membrane. Moist heat usually is much more effective than dry heat. There are different methods of using moist heat: boiling, autoclaving, pasteurization, and ultra-high temperature sterilization. Boiling is going to kill your vegetative cells. It will kill most of the viruses. It kills most of your protozoans. The trick here is how long do you boil? And a lot of people think, oh, I only need to boil 5 or 10 minutes. It depends on the elevation. The pressure changes as you increase in elevation. It makes a difference. Most people are familiar with, in the Celsius temperature scale, water boils at 100 degrees. That's what most people will tell you. That's true, but that's 100 degrees at sea level. For many years, I lived in New Mexico. Albuquerque is a mile high. It's 5,300 feet in elevation. We used to do an experiment there and would have the students boil water. So at a mile high, at least there, water boils not at 100 degrees C, but at 96. And you might say, okay, so it's 4 degrees difference. What does that translate to? That means you need to boil longer. So the higher you go up in elevation, the longer you have to boil to reach the same effectiveness. So that's why we're saying the boiling time is critical, and that's dependent upon your elevation. You want to boil long enough so that you are killing those vegetative cells. Endospores and some of the cysts and some viruses can survive the boiling. Autoclaving, this is what I was talking about a little bit ago. You're adding pressure. You're in a closed environment. Think about, if any of you do cooking, it's kind of like using a pressure cooker. So the boiling temperature increases as the pressure increases. So the minimum requirements for autoclaving are 15 minutes, 15 psi at 121 degrees C. Now, this is showing how the pressure increases in a closed environment, sealed environment, as the temperature increases. So this is showing a picture of an autoclave on the top, and they come in all different shapes and sizes. Personally, the one at the top, that might be great for, I don't know, maybe a dental office or something. In terms of microbiology research, that would be considered a very small autoclave. I have seen them as large as huge. We would use these rolling, it almost looks like a rolling cart with shelves on it, and you would roll the whole thing into it. It was probably about 4 feet in depth, and probably the diameter was about 2 to 3 feet. I mean, they're huge, some of them. So they come in all different sizes, but they all work relatively the same, as you can see in picture B. That you have your internal chamber, that's where you're going to put your supplies, whatever it is that you need to autoclave. It could be waste products, it could be media you want to sterilize. And you put it in there. There is a water supply that you have to basically turn on. You close the door, it is sealed. When you turn it on, then it heats it up, and as you can see, there are different temperature and pressure gauges. There's a jacket, what they call the jacket. This is that closed, open cavity area around where you have put your articles, and that's where the steam is. And so you heat up the steam, and it's going to be in that outer jacket that will heat the water up to produce the steam. There is going to be, like I say, pressure gauge. There's also, on all of them, there is going to be a safety valve so that if the pressure gets up too high, it can release that. When you run things through an autoclave, you typically will add something that you can see that did you reach that level for a long enough time to sterilize whatever your supplies were. And so there's different ways of doing this. Sometimes you can use these nice, what I would call fancy vials that you break substance and you look at a color. If it's yellow, basically you did not reach high enough temperature. If it's red, it means things that you autoclaved are sterile. They'll show this in reality. Most people I know, the thing that I've always used and most labs use, there's what we call autoclave tape. It looks like just regular masking tape, and you just rip off a piece and you stick it on your trays or on the bag, or you don't necessarily put it on every single, say, test tube and flask. Let's say you're autoclaving a rack of test tubes. Standard rack holds 40 test tubes. So you're not going to put a piece of tape on every single test tube. Just put it on the rack. Or if we often put trays in one tray, these plastic tubs, one holds 2 racks. So we put 2 racks of test tubes on that. We just put one piece of tape on that tub or on the tray. And what happens with the autoclave tape, if you did reach proper conditions and so things are sterile, black lines will appear on the tape if it's sterile, if you reach those conditions. So You just put a piece of tape on your, like I say, on the tub, on the rack, and then afterwards when you go to pull it out, look, do you have black lines? Okay, you're good to go. And that's usually a lot cheaper than those vials. Pasteurization is another form of moist heat that's often used for dairy products and juices. It's not sterilized. So if organisms can withstand high temperatures, they're still going to be there. But the idea is that you are killing the pathogens. Why don't you sterilize it? Sometimes sterilization process will alter the taste or the flavor. And so that's why they don't tend to do it for things like dairy products and juices. For pasteurization of milk, there's different methods of pasteurization: batch, flash, and ultra-high temperature. Ultra-high temperature is where you increase the temperature to 140 degrees Celsius for 1 to 3 seconds, and then you rapidly cool it. Those fluids then actually can be stored at room temperature. So this is showing some of the differences between the pasteurization. Flash pasteurization is 72 degrees C for 15 seconds. Notice that the higher temperature you go, the less time you have to go for. Dry heat. This can be used for materials that can't be sterilized with moist heat. It will denature the protein still. It will oxidize certain chemicals. Usually you have to go for a higher temperature for a longer time than you would with the moist heat. The ultimate of sterilization is incineration. Basically, you're going to burn it. And that's what we do in the lab oftentimes. Whenever you flame, like an inoculation loop, you hold it in the fire and you flame it. That's actually incineration. That is what you're doing. In terms of storing, you have refrigeration and freezing. This is going to decrease the microbial metabolism, so it decreases growth, decreases reproduction. Chemical reactions tend to go much slower when you decrease the temperature. If you freeze something, then liquid water is not available for the organisms to use. So refrigeration is going to not necessarily kill, but it's going to inhibit the growth of most of your pathogens. Now you take it out and put it at room temperature, and they could start growing again. There are some microorganisms that can multiply in the refrigerated foods. You've all seen that. Some fungi. Those strawberries look so good at the store, and you bought them, and you put them in your fridge. And maybe, you got sidetracked and you kind of forgot, and a week later you go to pull them out, and there's all these nice little white fuzzy stuff growing on them. That's fungi. They're able to grow in the fridge sometimes. Slow freezing is more effective than quick freezing. Some organisms can survive freezing, most of them cannot. Desiccation and lyophilization. Desiccation is drying out. You're removing the water, and then therefore, because all living organisms are dependent on water, if you remove the water, then that's going to prevent the growth of them. So that's one way of storing. Done that for years where people have dried out, dried fruits, etc. Lyophilization is a little bit different. It's a freeze drying method. We do this sometimes in the lab when you want to preserve, say, a microbial culture. If you want to preserve it for long term, you don't necessarily want to just take the culture and put it in the freezer, because when the water freezes, it forms ice crystals that can damage the cell membrane and cell wall. And so if you freeze dry it, it becomes like a powder. And so it prevents the damage from the ice crystals. It also takes up less space. So you can store a lot more in the same amount of space. You can, when you lyophilize something, you can store it for long periods of time. Oftentimes they will store these at like 70 below in the ultra freezer, and then when you remove it, you just rehydrate it. So this is an example of desiccation, like I say, drying. In this case, they're drying fruits as a means of preserving them. Sometimes you may have something you need to sterilize, but in this case, it's looking at this broth. You might have a particular, let's say, a protein in there that you're trying to sterilize. If you put it in the autoclave, that moist heat is going to disrupt the structure of the protein and destroy it. You want the functional protein. So how do you sterilize it? You can filter it through a membrane. So membrane you can get, you buy these, you buy them with different pore size. So you select one that would be, the pore size is smaller than any microorganisms that you suspect may be there. And then you would just pour into the apparatus. Usually you have it hooked up where it says to a vacuum pump. Oftentimes that's hooked up just to a water line. You turn the water line on, that forms a pump, and that's what's pulling the liquid through that filter. What you collect in the flask, that flask has already previously been sterilized. So what goes in there is sterile. In the picture on the right, under B, you can see the little holes on the blue membrane filter. Those are the pores. But notice the bacteria, in this case, is not able to pass through. And this is showing you the different pore size. And like I say, you can select which size you want to use, and depending on what type of organisms you're wanting to trap. Some people buy water filtration systems. I knew someone that used to do a lot of backpacking, and to make sure he had clean drinking water, he was so proud of himself that he had bought this filtration unit that was very lightweight that he could bring with him, so he could scoop water out of any water source, run it through this filter, and he would have clean drinking water, or so he thought. And he was so proud of himself because the pore size, when I asked him, I said, well, what pore size is it? He goes, oh, it's a 5. And I was like, that's not going to do you a whole lot of good. If you notice here, that might keep out some of your larger algae, animals, and fungi, but it's not going to keep any bacteria out. You need to go much, much smaller to keep bacteria out. A HEPA filter is a high efficiency particulate air filter. This is what we use in those biological safety cabinets. This is similar to what I showed you in the background of the picture of the person working in the BSL-4 lab. Sometimes you will work in these, we used to just call them the hoods, airflow hoods, or just the hood. There is a special way when you turn it on. The airflow, as you can see, is pulling things essentially away from the front. And there's a safety screen that can move down, and you just move your arms into the hood, do the work. You're working everything with your sample inside that hood. The idea is to prevent any potential contamination of you. You're trying to keep the microorganism and any other contaminants inside that hood. The airflow is flowing it down, and then it will pass through the filter before it's then released to the outside. That filter is a very fine pore, and it will keep the microorganisms, trap them, so they cannot be expended out, to the outside environment, but it's also a means of keeping you safe. Osmotic pressure. High concentration of salt or sugar in foods will help to inhibit the growth. Cells, if they are put in a solution that has a high concentration of salt or high concentration of sugar, that would be a hypertonic solution, they tend to lose water. Now, fungi have a better ability to survive than bacteria in a hypertonic solution. Radiation, ionizing radiation, things like gamma rays, some X-rays, This can disrupt some of the hydrogen bonding, such as what you see in DNA. And so it can be somewhat effective. Gamma rays are not that effective. They can penetrate, but they're not necessarily going to kill the microorganisms. X-rays would require a long time, so it's not, the time factor is just not practical. Electron beams are effective at killing microorganisms on a surface, but they don't penetrate well. So as you can see, there's plus and minuses to all of these. This is showing they have started using some ionizing radiation with certain, in this case, produce, certain items. Obviously, something like strawberries, which are highly susceptible to contamination because everyone, including fungi, like them, not just you. You can't autoclave them. That's going to totally destroy them. And so they have started using ionized radiation. It's a balance, though, because once again, you have to look at the pros and cons. Does it penetrate well? How long do you have to expose it in order for it to be effective? It is the problem with any type of soft fruit is that they tend to be more susceptible to not only bacterial but also fungal infection, and there's a post-harvest issue. There's a very limited time from when you harvest them to how long they will remain good, and you have the farmer obviously has to get it from his field to the market, and then from the store to your house. There's a very limited time frame to get that done. Non-ionizing radiation will also affect the structure of proteins and nucleic acids. Things such as UV light. UV light does not penetrate. It does not penetrate through glass, does not penetrate through plastic. So that could be a problem. However, there are some benefits to UV light. It can be very effective in disinfecting the air. If certain fluids, but it depends on what the fluid is in. It's very good at surface in disinfecting the surface of objects. So some labs, and I've actually worked in labs where they've done this, where they will have UV lights all around on the ceiling, especially along the walls. And the last person to leave, when you leave for the day, you shut off the regular lights and then you flip on the UV lights. And the UV lights are left on all night. And the first person to come back in the morning, when they come in, they shut off the UV light and then turn on the regular lights. And that is very effective in disinfecting the air and disinfecting the surface. Now, obviously, you should still be wiping down your bench tops when you're done. It was just kind of like an extra protective mechanism type thing. In the teaching labs, what we often do is we will have goggle cabinets where we provide goggles for students to wear while doing their lab exercises. And between each class, we put the goggles back into the case, we close it up, and then we turn it on. And what it's using is there's a UV light in there that then is used to disinfect the goggles between classes. So that way we can always provide students with goggles that have been disinfected. So UV light, no, it doesn't penetrate well, but it's good on surfaces. It's good in the air. So it has a purpose. This is just a chart that is summarizing these are all physical methods for trying to control the microbial growth. Chemical methods, this is going to affect the cell walls, the membrane, the proteins, or DNA. Depending on environmental conditions, the effect will vary. Some of these are more effective against envelope viruses and vegetative cells. You once again start throwing in endospores, and that's going to be a little bit tougher. Phenol and phenolics, these tend to disrupt proteins and disrupt cell membranes. They can be very effective when you've got a lot of organic material there. It does remain active for long periods of time. They can be used in healthcare settings. They're used in labs. One problem with them is the smell and possible side effects. So this is an example of, from a chemical standpoint, the structure, what the phenolics look like, usually ring structures. Alcohols are intermediate level disinfectants. They will denature proteins. They will disrupt that cell membrane. It'll be a little more effective than soap. However, a lot of people talk about their hand sanitizers, which tend to be very high in alcohol. It's going to dry your skin out a lot. Usually you will swab the skin with alcohol prior to any type of an injection. That's the little, you see them wipe the area. That's to remove most of the microorganisms that are on your skin in the area where an injection is going to be given. Or if they're going to, say, draw blood. Halogens are intermediate levels. They damage enzymes, which are proteins, by denaturing them. Iodine tablets, chlorine bleach treatments, bromine disinfection. These are all different examples of it. So you can see here, they're wiping an area on a hand that's apparently going to have surgery, and so they wipe that with beta iodine to remove the microorganisms on the surface of where they're obviously going to do an incision. You don't want to introduce anything into the body from along the incision site. Oxidizing agents, things such as peroxide, they will kill by oxidizing certain enzymes. These are high-level disinfectants and antiseptics. Hydrogen peroxide can disinfect and sterilize surfaces. However, you don't use it for deep wounds. Ozone can be used for treatment of drinking water. Surfactants. This includes soaps and detergents. They're good at rinsing off agents. One thing, if you're going to use soap or any type of surfactant, soap, detergent, washing, it's critical more the timing that you wash it for the proper length of time. During this recent COVID-19 pandemic, certainly, if nothing else, some of my friends in microbiology and I've said, and in the healthcare field in general, if nothing else, people hopefully are finally learning to wash their hands long enough. Just rinsing through water, 1, 2, 3, you're done, doesn't count. You need to be going for at least 20 seconds. So it's the timing that you're using as you are washing. These are low-level disinfectants. The detergent's going to help interfere with the cell membrane. This is some examples of some quaternary ammonia compounds. Heavy metals tend to denature proteins. They're low-level bacteriostatic and fungistatic agents, so it's going to inhibit. Then 1% silver nitrate is often used. Copper can control algae growth. This shows where they have put some heavy metal on a plate and notice that it inhibits the growth around it. Aldehydes. Some aldehydes, like glutaryaldehyde, can be used to disinfect and sterilize. Formalin can be used for disinfection and embalming. Some of these you do have to watch. Formaldehyde was used years ago for embalming. It is a carcinogen, so they've switched to a different form, which is called formalin. Some places you do need to check for proper disposal, whether that can be allowed to go down a drain or not. It varies by city ordinances. Different gas agents. If it's a closed environment, obviously you can use different gases to help sterilize items. Oftentimes the gases will denature proteins, which you're starting to see. That's a very common thread through all this. Denature the proteins, denature or disrupt the structure of the DNA. This can be used oftentimes in hospitals and dental offices. There are disadvantages, is that It has to be in a closed environment because it can be hazardous to people. Oftentimes, some of the gases used can be highly explosive, so you have to be very careful with that. It can be poisonous and potentially carcinogenic. Enzymes. There are some antimicrobial enzymes that different other organisms make to act against microorganisms. One example is your tears in humans. You contain an enzyme called lysozyme. Lysozyme is going to break down the cell wall of bacteria. So it's an antimicrobial agent that your body naturally makes. Every time you blink your eyes, those tears are going across the surface of your eyes, and it's a way of trying to help prevent penetration of any bacteria through your eyes. Now, sometimes we can use these also in the environment. Lysozyme can be used to reduce number of bacteria in cheese. And then you've got your antimicrobial drugs, antibiotics, semi-synthetic and synthetic chemicals that are typically used for treating disease. And this is a list of the chemical methods that are used for microbial control. There's different ways of evaluating how effective different disinfectants and antiseptics are. These change over time. They used to use what was known as a phenol coefficient. That's been replaced. They can use dilution test to see basically what you're wanting to do is find out at what concentration your particular chemical that you're looking at, say you're looking at chemical methods. So you've produced some new chemical. Hey, I think this is going to work. What you want to do is, number one, test to see if it's effective, and then also test at what concentration, because if you're going to be able to use it, you want to be able to prevent growth of microorganisms completely, not just little. You want to prevent the growth, but you need to know at what concentration. You don't want to use more than you don't want to use 10 times more than you have to. So you've got to have a test. And these are listing different ways that you can measure measure what's the lowest concentration that I can use for it to be effective. And that's what this is. All these different methods here are just different ways of testing that. If you only need to use, let's just say, 1 gram per milliliter. I'm just throwing numbers out here. If that would be sufficient, then you're not going to want to use 20 grams per milliliter. Why would you use that much higher concentration if one is effective? And so you've got to have ways of testing that. Development of resistant microorganisms. One of the things that we have to do is be very careful. If you use a lot of disinfecting chemicals and antiseptics, does that help to promote resistant microorganisms?